Hello, thank you very much. First, I would like to thank the IAAS and Professor Ela Shaked for organizing this conference, for inviting me to give this talk. So today is not going to be a pure scientific talk. I'm going to share with you my scientific path from my master to my postdoc. I'm going to share my scientific interest as well as some personal challenges. So Professor James Lovelock is the founder of the God hypothesis, suggested that life and their environment are highly interlinked. Plate tectonic creates the oceans, mountains, deep ocean vents, niches where life can evolve and diversify through time. On the other hand, the biology highly impacts global biogeochemical cycles. Perhaps the most notable example is the activity of this marine unicellular microscopic organism named phytoplankton. Since their evolution, they highly impact the surface of our planet. They are responsible for the accumulation of oxygen, and by taking up inorganic substances such as carbon dioxide, nitrate, phosphate, and trace element, and converting them into their cellular material, they redistribute these elements between the ocean atmosphere and the sediment. So I was first introduced to this magnificent organism during my master at the Weizmann Institute. And in the group of Professor Asaf Vardi and Ilan Cohen, I investigated the mechanism by which viruses of phytoplankton are emitted into the air, dispersed to distant places, and infect phytoplankton, healthy phytoplankton population in remote places. So during my master, I did a lot of laboratory work in using tools for molecular microbiology. I did a lot of field work. I was even participated in a research cruise in the North Atlantic. And I had a really great time during my master with great mentors and great groups. However, toward the end of my master, the continuation of my master's seems natural into a PhD. However, toward the end of my master, I felt that is not exactly what I wanted to do. I wanted to ask bigger questions about the way the biology and the environment interact. And I wanted to use more mathematical tools in order to ask this question. So during the literature review that I did, I came across a paper that really blew my mind. And this paper was conducted by my current postdoc advisor, by Professor Mick Follows from MIT. So they developed the Darwin model, which is an ecological model that simulates the growth of this organism in the global ocean, which is embedded in the MIT general circulation models. So they basically simulate the growth of the phytoplankton, which are growing, they are photosynthetic organisms. They grow, they uptake inorganic nutrient and trace elements, and they use up light in order to grow. I will show you a video. I don't know. Okay, so what is so amazing about this model, the color here is a different phytoplankton group, that they simulated large amount of phytoplankton groups and they distribute them evenly around the globe. And as they run the model forward in time, the biogeography of the phytoplankton emerged. And in a way, they simulated an evolutionary process of natural selection. And this is why it's called the Darwin model. And I really was excited about this model. And I decided that this is what I wanted to do. However, I didn't approach Mick Follows back then. First, I didn't feel comfortable. I didn't have self-confidence enough because I didn't know how to model. I was doing mostly experimental work in molecular microbiology. And also, I didn't even know how to program. So I didn't contact him back then. However, lucky enough, a new PI came to the Weizmann Institute. His name is Professor Itai Alevi. He's a geologist and computer scientist. He didn't do exactly what I wanted to do. However, what I liked about Itai is that he asks big questions and he used very quantitative tools in order to answer them. So I approached him with my ideas and I asked him whether he can take me to a PhD, and luckily he agreed, and I'm really grateful for him for that. However, in the first 2 years, I was really much frustrated. Sorry. Yeah, I had many ideas, but I didn't have a specific research question. I was mostly in front of the computer all day, and I had to read a lot of geology, which is a field that I was not really natural inclined to. So it was really difficult time in the beginning. But after maybe 2 years, I found a research question that was meaningful enough for me to answer. The question is concerning the Redfield ratio. So approximately 90 years ago, Alfred Redfield is a famous oceanographer. He discovered that the ratio of the inorganic nitrate and phosphate in the ocean is constant and equal to the ratio of these elements inside of the phytoplankton cells. And this is a great manifestation of how the environment and the biota interact. However, 90 years after, a lot of scientific and research scientific data accumulated and showed that it's not a constant and there are global variations in this ratio. So in my PhD, I asked what are the factors that control these variations, and also I asked how this ratio varied through the geologic past. So in the first chapter of my thesis, I found that these global variations in the Redfield ratio are mostly due to changes in phytoplankton community structure. So different phytoplankton groups in the lab have different elemental composition, and their biogeography dictates the variation in the elemental ratio. In the other chapter of my PhD, I developed a model of the carbon, oxygen, alkalinity, and the phosphorus, and I simulate what were the environmental conditions in the geologic past, and I found that the Redfield ratio was not a constant as usually assumed, but it was higher than today, and it slowly decreased to present day value due to several evolutionary and geologic events, like the evolution of land plants in the Devonian, and the formation and the composition of the supercontinent Pangaea in the Mesozoic. These events highly changed the nutrients in the ocean and the climatic conditions and led to a decrease in the Redfield ratio. Okay, so after my PhD, I felt confident enough to contact Mick Follows. I never met him in a conference. No one introduced us, but I just wrote him an email. Hi, my name is Shlomit. I really enjoy your research. Here is my CV, and I want to apply for a postdoc. And lucky enough, in the same day, he replied. He read my papers, and he was really happy to meet me. And he invited me for a postdoc position at MIT. So I was very much happy. However, I was pregnant back then, and I had to delay my arrival in 1.5 years. So during this time, I was doing a short postdoc with Dr. Yoav Lehan from Haifa University, where we study phytoplankton from remote sensing, which is a project that I really hope to continue. But in the last few minutes that I have, I will speak about what I'm doing now in my postdoc. So the first generation of models that are embedded in the general circulation model assumed a fixed elemental composition with a classical Michaelis-Menten uptake. The second generation includes viral pools of the nutrients inside of a cell. And what we're working on developing now is resolving the macromolecular composition of the cells inside of the phytoplankton. We have now reached a point that we have enough of physiological knowledge about the factors that govern these variations. And this is what we are focusing now. And this is very important to resolve the macromolecular composition because it's important to understand the energy and the material transfer in the marine food web. However, it's not so fair to give one name on top of these people because this is really a work of a lot of people from working in labs, measuring these processes, establishing protocols to measure these things in the field. And I'm a small part on this project, but I'm really happy to lead this modeling effort. So this is not only a technical question. I have a lot of scientific questions that I would like to answer. And I'd like to understand what are the global variations in the macromolecular composition, how many calories are being produced by the marine phytoplankton. They are the source of the whole marine food web, and it's very important to understand this. How is the macromolecular and elemental composition expected to vary under climate change scenarios? And in what way will variable stoichiometry affect the oxygen budget? Because we know different macromolecules consume different amount of oxygen. So I would like to also ask this question. So these are some initial results. We found that approximately 60% of the carbon that is being fixed by marine phytoplankton is fixed in the form of proteins, 16% is in the form of carbohydrates, and 80% in the form of lipids, and less in the form of nucleic acid. And there is a clear latitudinal trend in this macromolecular composition where proteins are mostly produced in the high latitudes, while lipids and carbohydrates are mostly produced in the gyres. Pleasantly, these results of macromolecular and elemental composition closely reflect global observations. So to sum up, I showed you today how in my master I study more molecular biology tools, and then in my PhD I developed 1D models and more cellular models. And now in my postdoc I develop in 3D models that simulate again the cellular physiology. And I really would like in the future to use everything that I acquire in order to ask how cellular processes in marine microorganisms, what is their global impact. And today when I walked around and I saw a lot of lecture and posters, I all the time I saw many labs here that measure kinetic rates and oxidation rate and a lot of questions that I really would like to collaborate in order to ask what is the global impact of these processes that you measure here in the lab. So with that, I would like to thank my postdoc advisor, Professor Mick Follows, and a lot of collaborators. I would like to thank my fellowships, Fulbright, Rothschild, and the Simons Foundation that I urge you to apply if you want to go for a postdoc. And we're going to discuss it more in the panel that is after this meeting. And I want to thank you for listening.